Hello, my name is Mason Cipollini with my partners Mackenna and Mayra Mendoza. This is the Luminescence lab for OPTI 471A.
Luminescence is the process where a molecule absorbs energy and then gives off light as it relaxes back down to the lower state. This excitation can happen through electronic or vibrational transitions, and what's important is the molecule is in thermal equilibrium when this happens. So basically the molecule gets excited, jumps to a higher energy level, and then releases the extra energy as a photon when it comes back down. The idea is the foundation for all the type of luminescence we studied in this lab. Fluorescence is one of the most common and fastest forms of luminescence. In fluorescence, the molecules absorb higher energy light like UV or blue light and then releases light as a longer wavelength. The lifetime is extremely short, just a few nanoseconds, so the emission looks it's instant to our eyes. Each dye has its own characteristic emission band based on its vibrational level. We also saw that fluorescence only happens when excitation light has enough energy. So when we tried using red light, none of the dyes glowed because the photons didn't have enough energy to excite them. For all of these plots, we used the same procedures. We placed each sample in a cuvette, measured absorption with white light source, and measured emission by exciting the samples with UV, blue, green, and red light. Each dye showed the absorption and emission peak we expected. The emission shapes stayed the same no matter the excitation color. The fluorescein only UV, blue, green produced real fluorescence. The red peak is just the laser line, which was expected. The minerals, detergent, and paper all showed strong blue fluorescence under UV, consistent with their known brighteners.
Phosphorescence is a type of luminescence that occurs when the material emits light of a higher wavelength than that which it absorbed. This effect occurs because phosphorescence involves a spin-forbidden transition. When the electron is vibrationally relaxing through the electronic energy levels, it may cross into an energy level triplet. The electronic energy levels are bound by the 4 quantum numbers, and without exterior radiation, the energy may become locked in by the spin. From this, phosphorescence can occur once an outside source provides enough energy to allow their transition to the ground state. That transition carries a very low probability, which results in a high lifetime. For this experiment, we first shined UV light onto the surface of the phosphorescent big star and measured the emission spectrum. Then, once we shined UV light onto the surface again, we attempted to measure the radiative power over time to find the star's lifetime. We were successful in capturing its emission spectrum, showing a high peak in the UV range and a smaller but wider peak in the 475 to 575 nanometer range. This is about what we expected, since it will emit light similar to the source, as well as a visibly green hue, which matches up with the second peak. However, we were unsuccessful in finding the lifetime of the star, as the silicon detector we were provided could not properly detect the UV laser.
Now we'll talk about chemiluminescence. Chemiluminescence happens when a chemical reaction directly produces light. Instead of needing an external light source like in fluorescence or phosphorescence, the reaction itself forms an excited product molecule that releases a photon as it relaxes back down. To observe this, we activated glow sticks of different colors and immediately collected their emission spectra with the spectrometer. Each stick solution was placed near the fiber input so we could capture the full emission curve right after mixing. We normalized the spectra so that each peak had the same scale, making it easier to compare brightness and wavelength differences across colors. As shown on the chart, the blue and violet glow sticks peaked around 450 nanometers, while green was centered around 520 nanometers, and yellow and orange shifted further into the red. These distinct peaks indicate that a few different fluorescent dyes are reused across multiple colors. Blue and violet share one dye, while green, yellow, and orange come from others or combinations. The spectra match the expected emission ranges for common glow stick dyes, confirming that the visible color directly reflects the dye's emission wavelength produced by the chemical reaction.
In this lab, we liked seeing each of the luminescent effects and gaining a better understanding of these concepts. I especially liked learning about phosphorescence, as I routinely wear a phosphorescent necklace, and I'm glad to better understand how it works. What we didn't like was our inability to collect the full results for this lab due to the faulty photodiode we had in our section.